हेलो। 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 किधर जा रहा है? हेलो। 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 हेलो।
ये मैं इधर खड़ी रहेगी तो चलेगा ना, उसे कुछ डिस्टर्ब नहीं करना।
Hello. A very good morning to all of you. A warm welcome to one and all who have gathered here this morning to attend the Dr. A.M. Bhagwat Memorial Lecture under the aegis of RJC Lecture Series 3.1, being hosted by the Department of Biological Sciences in collaboration with Association of Teachers in Biological Sciences. At the very outset, may I invite Director Madam Dr. Usha Mukundan Ma'am to kindly welcome the gathering. Madam.
Good afternoon, everyone. Respected Dr. Shashi Menon. There are many adjectives which I can add for him, but I would say in one word, an excellent human. On behalf of Hindi Vidya Prachar Samitis, Ram Niranjan Niranwala College, I extend a very warm welcome to you on this very special occasion when we are remembering our very beloved teacher, Dr. Ashok M. Bhagwat. Respected Principal, Dr. Himanshu Davda, Vice Principal, Major Praveen Nayak, all teacher colleagues from Department of Zoology and Botany, and you very dear students. It's a occasion which is also very emotional, especially for some of us, because we were taught by Bhagwat. And whatever we are today is because of some very excellent teachers that we have in our Department of Biological Sciences. In University of Mumbai, I think Junyanwala College is very unique that botany and zoology, they coexist, due to which there are many things that we can share and care for each other. Bhagwat sir was an excellent human being, a teacher par excellence, an academician, very good researcher. And if you look at him, his personality was very unique. And some of us were very fortunate to have been trained by him. And he always firmly believed that a good teacher should also be a good researcher. He did not speak a lot, but that one sentence would convey many things. And when we were students, we would always go and sit on the first bench to just admire him, the way he would teach the subject, the lucid language that he will use. And in that era, in 1970s, to go abroad to USA in a Fulbright scholarship has its own meaning. Because nowadays, I think every family, you have somebody who is abroad. But in those times, you did not have teachers who would avail of this occasion and would travel to another country to just gain the expertise. And when he came back, he was rejuvenated. He has guided many students and they occupy very prestigious positions, both in India and abroad. And I'm very sure one of his students, Dr. Naresh Chirmule, who is in US, and you will also get the good opportunity to meet him sometime, would be listening to this session. And I'm very sure each and every student of Bhagwat sir will go back in the memory lane, go back to the room number 11 or 12, go back to the inter-science or BSc lectures and remember what a good time we had. And I'm very sure the same time also continues today when we as teachers are speaking to you all in some session or during any lecture. Bhagwat sir guided students in different areas and he also brought a good amalgamation of combining botany and zoology together. He was an expert in biochemistry. He did very good lab work, but he also believed in field work. So there were many students who have got their PhD in field work. And go back and see, there is Dr. Yajvendra Dhala, who is the director of the institute in Dehradun, which is specializing in wildlife. And he would share and care for the students. Whatever opportunities come, he would tell the students about those opportunities and open up windows and doors for them. He was also the controller of examination for some time. He worked in the examination committee very closely. He was a very big support for our guide and mentor, Dr. Karmakar, who occupied the position of the principal for a very long time. As you all know, that when you are a principal, you have to do a lot of multitasking and you need a big team, which will stand by you all the time. And Dr. Bhagwat provided that very big support for Dr. Karmakar, because he had the capacity to maintain confidentiality. He had the capacity to listen, because listening is also a very big skill which people need to acquire. So he can listen, he was a very good listener. And at the end of the day, he would come out with some very good solutions, because sometimes there are decisions to be taken. And when you are deciding, you would like somebody else also to come with you. Any new innovation, whether it is in terms of coding the answer books or when computers came in early or late 70s, he was the first one to take that and also train others in that area. And then later on, he moved to high-tech research, whether it was in the field of molecular biology. He was also the vice principal of the college. And then he all decided that he would like to do full-time research. And he joined the C.V. Patel Institute in Mitibai and did research for more than a decade. He took along with him mentor, guide, Dr. S.M. Karmakar after his retirement, and both of them enjoyed a very long tenure with each other just doing research. Today, this lecture is dedicated to Dr. A.M. Bhagwat in his memory, whom we lost 2 years back. I feel just 2 years, and Janvi tells me 5 years, but it feels that only recently we have lost him. And in his memory, the best way to remember a good teacher is to start a memorial lecture. I'm very sure many young teachers who are sitting here, they will carry on this legacy. It will not be just one lecture a year and you end it. It will go on till you students, someone will take the baton and continue with this lecture series. So this year we are starting 2 lecture series, one dedicated to Dr. A.M. Bhagwat and one dedicated to Dr. S.M. Karmakar. Both excellent teachers, very good mentors, and in their memory, the only way that we can pay homage is to conduct lecture series, educate the young generation so that our young generation also take the legacy forward. On this occasion, I'm very thankful to Dr. Sasi Menon, a very good friend of ours, for accepting the invitation for this, delivering this first section, and I'm very sure all of you are going to faint after you listen to him, because you will think, oh my God, how can somebody deliver such a beautiful session? And the depth of knowledge that he has, and he's also one person who believes in sharing information with others. It's a very good research. He has taken the research beyond the horizon of classroom, and more about it, you'll be so listening. I'm extremely thankful to you, Dr. Sasi Menon, because the Association for Teachers in Biological Sciences have also joined hands with us for this memorial lecture series. So during the COVID period, we had RJC lecture series, and for you students, all of them are available on college websites. You can go, and some of them are excellent. Lot of information you can get, and now we begin with a new series this academic year and hope to have at least minimum 4 lectures in any academic year. So with the support of ATBS, I'm very sure we will be able to have some excellent speakers who can come and interact with our students and provide them with the required guidance. So thank you very much, and on behalf of the college and on my own behalf, on behalf of the management, I extend a very, very warm welcome to each one of you and have a very enjoyable session. But please also make it very interactive. You can ask question answers because Shashi has many things above his sleeves or under his sleeves. So I'm very sure he will share some of that information with you students because you are the youth power. So thank you so much for giving me this opportunity. And I also look forward, but maybe I'll see on the YouTube. Thank you so much.
Thank you, Usha Madam, for the address. Today, our guest speaker, as ma'am has already mentioned, Dr. SASIKUMAR Menon, sir, is a director, Institute of Advanced Research in Interdisciplinary Sciences, TDM Lab, and is an associate professor from Pharma Analytical Sciences, Kaushal Kendra, Ram Narayan Ruya College, Mumbai. Sir, we are indeed honored and privileged to have you with us this morning. May I request Dr. Usha Mukundan, Director, Hindi Vidya Prachar Samiti, and our principal, Dr. Himanshu Davda, sir, to felicitate our guest speaker for today's session.
Yes,
Thank you. I'm also being privileged to introduce our guest for this morning, Dr. SASIKUMAR Menon, sir. He is MSc PhD and he is a director and also associate professor. Sir has been teaching in Ram Narayan Ruya College since August 1983 in the Department of Zoology. He is a recognized research guide under the University of Mumbai, and sir has guided 23 students for PhD and 2 for MSc. Friends, the list is never ending. Sir is also having 3 students who are currently being supervised. So I think, sir, you will be reaching 26 this year. Okay, congratulations, sir. And many more to come, obviously. The research interest, his research interest in the field of drug toxicology, herbal drugs, drug addiction, action, reproductive physiology, male contraceptive, ecology and biodiversity conservation. Sir is also a principal investigator for more than 350 drug trials in human. My God, that's a big number, sir. He has more than 60 trials on cosmeceuticals and nutraceuticals in human, and more than 150 drug toxicological studies in animals. He is also a co-investigator of projects funded by DBT, DST, and Ayush, Ministry of Health, Government of India. To his credit, sir has also got the patent on herbal mosquito repellent, which has been granted, and he is a reviewer in several national and international research publications. He has got more than 100 research publications in national and international journals, and has a edge index of 19 and 1,330 citations. My goodness, sir, I think that we always retire by the time we reach this number, but we have already done it. And we are also lucky to quote here that sir is also a member of the BOS of Department of Zoology. Thank you very much, sir, for accepting the invitation and being here with us this morning. Friends, Bhagwat sir was born to teach, to inspire, to change people, and to never give up, even when he faced with challenges that seems impossible. A great teacher affects the eternity. He can never tell where his influences will stop. Even today, the teachings of Bhagwat sir continues to influence many souls, and we are indeed privileged to have MENON sir who continues the legacy of being good teachers. A pioneer in the field of science and an extremely excellent teacher himself, I invite MENON sir to deliver his talk on parasites and evolutionary enigma. Sir, over to you.
ये कम ही कर दूंगा हमारे लिसनर भी है वो अच्छी थी थैंक यू Good afternoon. Thank you, Usha madam. Thank you, Linda madam. I feel really humble today, frankly, from heart, because I am standing here in memory of one of my favorite teachers. So I didn't belong to this college, but he was my teacher during my MSc. And as a teacher, only one thing I'll tell you. He used to uncover the syllabus. That's what he used to do in the class. And he's the one who excited us to read more beyond what is being taught, beyond what is there in the syllabus. And I'll tell you all the inquisitiveness I have today, I should credit some part of it to his teachings and his, his encouragement to read more. I'll tell you, after listening to his first lecture, I went and brought my own copy of Biochemistry by Leninger at that time. And it was not available in Mumbai. I got the copy from Pune. So that's how he excited students. And he gave that way of looking at some problems and trying to find out why are they happening. And what could be the reason that these things are happening in nature, and can you find some solution? So this topic that I took up today to talk to you is based on that. Because parasites, we normally just don't bother. It's almost the most difficult topic because most of the things you have to by heart. And all these stages of its development and the intermediate host and definitive host and so on, treatment and symptoms. That's the end of it. But I will make you think of parasites very differently from today. That's what I have written it on the title. They are an evolutionary enigma. There are lots of things we don't know. How they came onto this earth and what are their lifestyles, how they affect the ecology of their host species. So that's what I'm going to cover with a lot of examples, and I'm sure you will enjoy. And being that it is open to a crowd with a spectrum of experience in academics, I have tried to make it very simple. I'll not go into the technicalities of all those things that I'm going to talk. But if I can kindle some kind of a spark of excitement in all of you, that I think this lecture has done the work, and I'm sure Bhagwat sir would love to have that response from his audience. So saying that, let me start. Okay. I will take some time to, get my speed up because I was not well till yesterday. So give me some time. So excuse me for that. So, I always tell students and everybody to look at organisms as a bundle of adaptations. That's how you should be looking at organism and try to understand it. So when you look at the organisms from a bundle of adaptations, what you see is that you will find that the animal has a lot of adaptations. Many of it is beneficial for its survival in a habitat where it is found, and that helps it to maximize the utilization of resources. Now, what does it end up in? Because of that maximization of utilization, it increases its survivability. And with this, it also reduces the interspecific competition and also gives better reproductive success, which will lead to the species sustainability because it transfers more genes to the next generation. So this is how you all know about an organism, and this is how we talk about organisms, but we never look at it in totality. So you need to have an ecological perspective to any organism when you look at it, study it, understand it, and also an evolutionary perspective. So when you link these things together, then you realize that an organism is so important in its ecological role, its niche, the habitat, and the entire ecological food cycle, energy cycles, and everything. That's how you encompass all these interactions of an organism as it's surviving in the habitat where it is found. So in that sense, if you look at parasites, we have undermined them totally. If you look at a food web, you hardly realize that you always talk about the producers, consumers, and parasites are a small portion. And then it really, you make if you look at that food pyramid, you see, itna chota hai, parasites are hardly any percent, which is not true. So we actually are grossly misinterpreting the parasites. Why? First and foremost, if you look at a food cycle or energy cycle or a food web or even a pyramid, you will find the diversity of the species of parasites is grossly very small. That's not correct. In nature, they are much, much wider. So how we miss that diversity? And most of the parasites have multiple hosts. And please remember, when I say multiple hosts, all these different hosts come from different food cycles. They are related to different food webs. And therefore, a parasite can not only influence a single host, because it is also having multiple hosts, it can influence multiple food webs and food cycles. This is the interesting aspect of the parasites, and that's why they are sustainable. Now, they use a very limited quantity of energy. So therefore, to understand them and their influence on the food cycle is also very difficult. So the latest ecological principles which we apply, mathematical models that we apply, look at parasites from the energy transfer point of view, not from their biomass, not from their number, but actually from the energy transfer. And that is when we give little better justice to the parasites in the food cycle. Now, the parasites colonize a host species and they are highly connected. They preferentially colonize only those host species, and those which have multiple host species, they are normally selecting the host species which are very close together. Means the species are related. We'll talk about those a little more later. Now, one thing you must understand about a parasite, it is never in the benefit of a host, benefit of the parasite, that it will exterminate the host totally. You saw that happening in COVID. There were so many people getting infected and people thought that gone, everybody will go. It will never happen because then the parasite also will not be there. So they have a strategy by which the host is sustained to survive. And this is the beauty of their evolution. We say parasites co-evolve with species, But we really didn't know, still even today, we don't know explanation to talk. How did they get those adaptations and those changes in their physiology, anatomy, and so many other things in genetic evolutions that has happened in them, we can't answer. There is no answer. So that is why, to me, they are still an enigma. Now let's look at some very interesting examples. As I told you, parasites are products of co-evolution. So they are designed to sustain by economizing the investment. Now, when I say investment, you all know parasites generally don't have any digestive system there. They don't have any major circulatory systems. Everything they get ready-made. So a lot of things they have not there, but they are all very nicely adapted to reproduce. This is one of the most important things about their existence itself. But they have a very significant impact on the ecological food chains. Now let's see how. So I'll take one example. One example is the Sessions trematode. It's like a liver fluke. It's a trematode, it's a parasite. Now what's the importance of this and the food cycle? You just see this interesting example. Now the Sessions trematode actually starts its life cycle with the snail. It's its first intermediate host. And then after it has developed into a certain stage, it comes out of the snail and then slowly get into a frog or a toad tadpole. And then it remains inside the tadpole till the tadpole has metamorphosized into an adult. The parasite is still there, and then it gets into the bird. That's his final definitive host. So the bird, when it feeds on the frog, the parasite gets into the bird. It's a very simple cycle, but this is not where the interesting things come. Now let me tell you how it actually completes this cycle. It starts from the snail, as I told you, because
recording stop again. Recording stop हुआ. क्लिक नहीं हो रहा क्या रिकॉर्ड बटन यहाँ पे? ये ओनली कंप्यूटर हो जाएगा. कंप्यूटर पे हो.
okay, sorry for that. So it gets into the, I'm not going to the detail of those stages of development, that's not the part of this lecture, but it gets into the snail, and after that, oh my God, ये वापस बंद हो.
अच्छा वो आपका पीपी है.
Okay. And then the cercarial larva of these parasite gets into the tadpole. Now this is where the enigma comes in, the mystery. The larva is in the stage of its metamorphosis. So it is developing its leg. And this parasite goes and sits very close to that spot where the limbs are getting developed. And then it secretes some chemicals. Please remember, I am talking about a trematode which has gone into a vertebrate. And the trematode is able to dictate the development of the leg of the tadpole by sending some chemical signals. So the trematode knows signals of the vertebrate. See the cross evolutionary relationship that has been established. This is something quite mysterious. How did it happen? Now, what happens is, this is not the fun. The tadpole grows the leg properly. But this fellow dictates that additional legs will be developed. These are called supernumerary legs. Actually, the bones are there, hardly any muscles, with a cover of skin. So you can see in that bigger photo, the frog gets multiple limbs, hind limbs. Now, what is the benefit of all this to the parasite? Is that the frog with these multiple limbs gets easily eaten by the bird. So actually, the parasite is supplying easy food to the bird, and in that process, completing its life cycle much faster. So that it gets into the bird much easily. Now, this happened, these things are just about 10 years old, these new findings, because people started working on it, molecular basis studies have been done. They found, this parasite is also very intelligent, in inverted commas, of course. It will first infest frog populations in a part of a lake, and then it shifts its infestation to another part of the lake, and then it changes its location so that there are enough frog populations sustaining in the lake so that it can go on infecting the frogs and thereby getting into the bird. Now, this is what I call co-evolutionary enigma. You can't explain all these things. Need to know why. And now the interesting thing is that if we can understand what are the chemical signals that are being released by the parasite, we can actually help development of limbs in people who are wound healing, where the wounds are not healing and things are not getting regenerated. These peptides or any of those chemicals can help us to deal with that. This is just one part of those things. Now let's look at this one. This is our friendly neighborhood parasite, the dodder plant, Cuscuta. You all must have seen it growing on the tree like this, correct? And you must have studied it sends Hostoria and so on, and it feeds, it pulls all the nutrients, it doesn't have chlorophyll, so everything can be do. But have you all really thought that how does it do it? Okay, now if I show you this TS of the Hostoria entering the host, you can see from the Hostoria, you can see the can you see the from the Hostoria, you can see the hyphae coming out, those small strand-like thing, and those hyphae go deep into the vascular bundles, and that is how the food is sent back to the Cuscuta. Okay. Now, if you go a little more closely, if you do an electron microscopic section of this and look at it, and you see that the red color that is shown, the red highlight of those the cell wall is of the parasite, and the blue is of the host plant. And you can see at one end the pink color, the red color has almost totally penetrated the blue cell wall. And that is how the link is taking place. Now, please remember here one thing. Two different unrelated species are setting up a cellular connection. Absolutely two genetically independent cell lines are getting a connection and sustaining it. So what are those chemical signals that work on these things? And how is it sustained? This is the question that we need to find answers. Okay. Now let's go a little deeper into this relationship. This is how the Hostoria sends in hyphae and then how the connection is settled. So first in scenario one, what happens is the Cuscuta sends some signals, which are shown here as yellow dots. And then in response to it, the plant sends some signals, those green ones. Then the Cuscuta sends additional signals, which are those white triangles. Now, due to this interaction of signals, the Hostoria enters and the host cell wall, you can see, is opening up. And then finally, a connection is made. Now, this connection between the host and the parasite is something that is not clearly understood. How can this connection happen between two absolutely different species? Okay. So now, so this is how it, just I explained to you. And then there is one question. Now, this must be happening all across the point where the host and the parasite has come in contact, but only at one or two points, the full connection is made. So there is actually a competition happening at every site, and then one or two sites, the process completes very fast, and that is from where the cuscuta takes up the food. But then how does the cuscuta know that these are the points where the connections are going to be made? We still don't know how the cuscuta comes to know about it. But the plant is also smart. The host plant is also smart. It has its own mechanism to resist it. Let's see how that is done. So this connection is what is called as the interspecific plasmodesmata, and that is very important. Interspecific. You can make plasmodesmata between cells very easily. But this is interspecific, and that is happening in nature. Now, you all know that the cuscuta goes on any plant. There are many species. It is a variety of hosts that it attacks. And actually, the smell of the host plant is what attracts the cuscuta spores to come and fall and germinate. Actually, volatile terpenoids are the main cause. And why this is very important is because the spores have hardly any nutrients. They survive only for 1 or 2 days. Within that 1 or 2 days, the spores have to find a proper host. And then only it will survive, and then you produce a new cuscuta plant. So for it, it is very important. So it uses these chemical signals to target the right host. Now please see the requirement of the host plant. It's just a simple vine, but it's doing all these things to get itself sustained in that habitat. Now here are the two photographs of the left-hand side. One is a susceptible one where the Haustoria has entered, and you can see the enlarged portion where the hyphae is also seen very clearly in blue color. And on the right side is a resistant one where the Haustoria is not able to penetrate and send the hyphae. They are all stopped there. And you can see at the end where the Haustoria has come in touch with the host, there is very dark staining. And that is because of lignin. At those positions where the host has come in contact with the parasite, the cells get lignified. And therefore, penetration does not happen. This is the way the host plant fights the entry of the Haustoria from the cuscuta. And today, because of molecular biology, we know a lot of things about this cascade. You can see there are receptor cells which identify the secretion coming from the parasite. And as soon as that is detected, a cascade of biochemical pathways are triggered, and then lignin is secreted, and then the resistance takes place. So that is how the resist. So you can see plant and the host and parasites are always in competition. One trying to beat the other to infest it, and of course the host plant trying to prevent this infestation. And this is evolving. Even today it is evolving. Now here is a very interesting thing. Now I am going to go a little more. These are very simple things. Now here it is going a little more in deep. Changes that be happening in the host plant is going to be absolutely, it will surprise all of you what the parasite is doing to the host. Now here is a parasite which is called as the horsehair worms. Obviously, because you can see on the hand, they look like the horsehair. Now these horsehair worms, they infest the cricket. It's an insect, carnivorous insect. So it infests the cricket. Now, how does it complete its life cycle? Now, this parasite has both terrestrial as well as aquatic part in its life cycle. So the sexual individuals of these parasites are aquatic. So their entire sexual reproduction takes place in the water, and then the eggs are laid, and the eggs convert into larvae, and the larvae then forms endless chironomous larvae. You all must have used in your lab the midge fly, the larvae of the midge fly. So these larvae of this parasite, they enter the chironomous larvae and form cysts within the chironomous larvae. Now this chironomous larvae then of course metamorphoses into the midge fly. That means there is no development inside the chironomous larvae. It is just a reservoir. Okay, it is just carrying all the eggs in the cyst form, the larvae in the cyst form, and the adult is also carrying this parasite in its cyst form. And therefore, since they are reservoirs, they are called as the parenteric vectors or the parenteric hosts. Now, the crickets, they feed on the midge fly. So now see what happens. The cricket will feed on the midge fly, and of course the cyst gets into the cricket. Now is the fun. Immediately, as soon as the cyst becomes adults or the cysts start entering and they become active in the cricket, the total behavior of the cricket changes. It starts loving water. And normally these things happen at the small forest around lakes and streams. And as you know, if you look into the water, there is bright light due to the reflection coming from the water. And the crickets are attracted to that bright light. They all start, those infected crickets, can you imagine, they don't have any control on their behavior. They just go into the water and commit suicide. They drown. And the larvae comes out and they all become adults again. So the cycle is repeated. So the parasite is secreting some chemicals which is changing the activity of the brain of the cricket and changes its hatred to water as its love and actually incites it to commit suicide. It dies. Actually goes into the water and dies. Now, even in our case, when schizophrenia patients are given some drugs, one of the side effects mentioned on those schizophrenia drugs is suicidal tendencies. And there are cases where the patients have just walked out, gone into the balcony and jumped down. So these crickets, imagine they lose their fear for water. So similarly, those patients lose fear for height. And they are just ready to do anything. So same thing happens in this parasite secreted chemicals in the brain of the cricket. See the correlation. And who is doing it? A small worm. Okay. This is another interesting parasitism. This is the gel wasp. Of course, it's a wasp from Hymenoptera group. And then we are looking at the host, that's your cockroach. Now, how does this life cycle of this wasp go? The wasp causes hypokinesia in the cockroach. That means it loses all its motor activity. The cockroach becomes like a nicely drunk fellow. He will go anywhere he wants, wherever he is pulled, he will go there. Now, how is the larvae, how is the wasp able to do it? Very interesting. Now, why the wasp is doing all this also, I'll tell you. But it starts like this. The wasp actually catches a cockroach, a live cockroach, and goes around the cockroach and stings the cockroach near the head. Now, you all have done dissection of earthworm. So, in the cockroach also, there is a nerve ring. So, there is a sub-esophageal ganglion and a supraesophageal ganglion, and the esophagus going through it. The wasp gives a sting to both these brains, both these ganglia, and the cockroach becomes nicely tired and timid. And then the cockroach is pulled by the wasp to his nest. And then the wasp lays a single egg on the cockroach, and the cockroach is still live. And then the larvae enters the cockroach, feeds on its entire body from inside, and then comes out as a new wasp. Now, if you don't believe this, I'll show you a video. Now, here are the supraesophageal ganglion and the subesophageal ganglion. Now, it's not always that the wasp is successful, please remember, because it has to target the two ganglia in short of time. The cockroach also starts to resist. It hits its legs and so on. Sometimes the wasp is hit and thrown away. But despite that, the wasp is able to do this and make the cockroach absolutely under its command. Now, what happens actually? So the first sting is given onto the subesophageal ganglion, and therefore what happens is it stops the thoracic ganglion function. So that cockroach's ability to run is cut off. And then because the thoracic ganglion is stopped, it also cuts off the other ganglia, that is the neurons of G and H, which also helps in movement of the legs under the control of the supraesophageal ganglion. So the cockroach is absolutely unable to control its legs, but it is able to walk. If pulled, it will go there. If pushed, pulled back, it will walk back, go to the side, wherever you want, but it cannot decide where to go. So the cockroach reaches in this stage. Then there is additional thing what this injection does, which the wasp gives. It also cuts off its escape tendency, because its antenna signals are blocked and its cerci signals are also blocked. You can imagine that with 2 injections, the wasp is able to make this cockroach absolutely a zombie. Now, see this video. It's got gamma aminobutyric acid in its venom. This is all done in a lab. So it's very beautifully done by the professors who worked on these wasp and the cockroach. See, the cockroach has become very timid and it starts cleaning its antennae, which is an indication that it has lost senses of its antennae and the cerci. So the wasp waits beside. ये सब ठीक होने दो, बाद में दिखाता हूँ तुम. And see, he waits and goes around, and now see what it will do. It's the cockroach is left there, and it knows cockroach will not go anywhere because its venom has started acting. It goes and searches for a nest, and then it pulls the cockroach. See, the cockroach is unable to make any resistance, and it cuts its antennae and starts to drain its blood. Thoda sa, because it's like a pipe. So the hemolymph flows into the wasp. Now it is pulling it to its nest. And then it lays the egg on the side of its leg. And then it will close the nest by putting all the gravel. And you can see the nest, 1 egg is put. So it's so confident that एक egg बस हो गया, खाना बहुत है, my young one will come out. See, the cockroach is still live. The larvae enters through the body wall and enters the body and starts eating inside and surviving and developing. Now, please remember the venom. Now you can imagine, this is the cockroach can get infected by bacteria and virus, so it can start rotting. It doesn't, because the venom of the adult, where the wasp has some antibiotic, and the larvae also secrete some antibiotic peptides, so that kuch nahi hota hai. Now these are very important peptides for us. Nothing happens to the cockroach, and then of course the larva comes out. So you can see this very beautiful work done by the Israeli scientists, and this beautifully made video. You can really see what it works.
But how does the wasp understand that the sun is so facially angular is here only?
Now that's the enigma which I was talking. There is no training. It knows and it does that in first shot. So even a young wasp will do the attack similarly like the adult. No, there's no training required. So imagine how much of evolution has happened, adaptations has happened. Now this is another example where the lancet liver, you all have learned about the liver fluke. It also has a similar thing. Now the liver fluke definitely hosts a cattle, but sometimes it can also come into the human being. So it also starts from its soil, and then snail, as you all know, I will not go to the detail of this. And from the snail, it goes into the cerci. Now these small larvae of this liver fluke, they form small globules with the slime of the snail. So these small globules are picked up by ants. Now see what happens to the ant. Moment the larvae has entered the ant's behavior changes. As soon as the sundown, that means the light has gone down and the sun is setting, the ants goes out of the nest. It starts walking out of the nest, goes onto a grass blade. Okay, I'm sorry, in the morning. As soon as the sun rises, the ant starts changing its behavior. It goes out of the nest, climbs a grass blade, and as it reaches the tip of the grass blade, as the photograph shows, it puts its pincers to the grass blade and remains there till it is eaten by a herbivore. If it is not eaten, then as the sundown comes, the ant changes its behavior. It removes its pincers, comes down the grass blade, goes to its nest and remains normal. Next day morning again it will do, till it is eaten by the herbivore. This is all because of the parasite which is giving signals to its brain to change its behavior. And people have seen that an ant can do this for at least 4 days continuously, and then it dies because there is nothing inside its body. Most of it has been eaten by the parasite. And that is how it is getting collected into the... Now here is a very distinct, different adaptation of the parasite. As soon as the larvae of this liver fluke enter into the ant, one of them races into the brain. And moment it attaches to the brain, it sends some very strong signals. Moment that signal is received, the other parasites, they all run into the abdomen. So you have seen the abdomen is full with parasites, and the brain there is one parasite. So if you take a TS of the brain, you can actually see the parasite sitting there, the red one, which is shown in the picture. So this is another thing, that within the larvae also there is communication. All of them don't go and go, only one will go to the cerebrum, that is the main ganglia, and then changes the behavior of the ant, where rest of them are just sitting tight. Abhi ho jayega. We will reach into the herbivore. So just see the adaptation. Now, these are things which we just cannot explain so easily, but they are all happening. This is another interesting thing of a fungus attacking an insect. Now there are many species of fungus which feeds on insects, and they are all gone into the classification of entomopathogenic fungus. Now what this fungus does is, it is in the soil. The spores are in the soil. Now the cicada is an insect, and many of the bugs, they actually have their larval cycles within the soil. So when they are in the soil, they come in contact with the spores, and the spores are inside, and the spores then develop into hyphae inside the insect. Here it is the cicada. And the hyphae starts feeding on the internal organs of the cicada. So much so that most of the abdominal organs everything is eaten. The body looks hollow, but the insect is walking. So the hyphae don't kill the main neuronal connection. And then what happens is, so within about the fungus spares a vital organ of the cicada, and it changes the behavior of the cicada. Now what happens is, I'll give you the example of the male. It can infect both male and the female. The males, when they get infected, they start behaving like the female. One of the behavior is that they sit and flutter their wings. So the other males are attracted towards this pseudo female. And when they come close to this, its abdomen is full of the spores of the fungus. That cicada is also getting infected. So one infected cicada will infect so many other cicadas as they come close to each other. And then finally, the cicada falls to the ground and the hyphae starts completing its life cycle. So this is now in Himalayas, there is one caterpillar which is attacked by a fungus. And this caterpillar, after attacked by the fungus, comes out of the soil and stands erect. And people collect that and are sold in the market very costly as aphrodisiacs. This is one of the biggest, trade that is happening in Nepal. They are highly in demand in China and many of the Southeast Asian countries. We have a good business going on in Uttarakhand and all the other upper reaches of Himalayas of this. So we also have a similar fungus which attacks the caterpillar. Now, what has been found is that these fungus produces some psychotropic substances similar to opioids, and they change the behavior of the larvae. Probably the same psychotropic substance, when we consume, we also change our activity and become hyperactive. And that is why these people use it as aphrodisiac. So they do contain some bioactive chemicals which even affect our behavior. Same thing, if you remember, rabies virus also does the same thing. It changes the behavior of the patient who is infested with rabies, or the animal which is infested with the rabies. Why? Because the rabies virus sits in the throat region. And therefore, the infected animal, whether human being also, becomes hydrophobic, just hates water, and they don't swallow. And that is why their saliva always comes out. And that is full of this virus. So the virus wants this animal to bite other animals so that the virus is transmitted to other hosts. So this is again a behavioral change brought in by a parasite which also infects us. So this is what I was trying to tell you. A virus knows some neuronal chemicals which act in our body, and it has got this adaptation of synthesizing it and secreting it once it has infected our body. So when there is a change in the predator and prey, there is always a defense mechanism. So this cycle continuously happens in nature. But how does it change the food cycle? I'll give you, there are two ways. One is a direct way and the other is the indirect. I'll just give you the names and you should know how these things happen. This is the bighorn sheep of the Northern America, and it's predated by a cat. I mean, it's the mountain lion. Now, these sheep also get infected by mites. That means their skin gets attacked like the dogs. Our pets also get attacks of mites. But here there is a difference. When they get attacked by the mites, their hearing is impaired. They can't hear. And because they can't hear, they are killed much more by the mountain lion. Now here what happens is, because there is infection of the mite, a parasite, the food supply to the mountain lion is increased. So the food cycle gets completely biased towards more of bighorn sheep being eaten by the mountain lions. So this is called as the consumptive trait mediated indirect interaction. Now what we just saw of the horseshoe, the horsehair and the cricket is a non-consumptive one. Now here the relation is very different. because the cricket is available, lots of them are dying in the water and jumping and dying in the water. The fish starts feeding on them and more. So now their diet is shifted to crickets. And therefore, other predators, other individuals in the lake are not predated upon, and they cause a huge loss of algae because they attack the algae. So if the crickets were not there, the algae is also eaten. So the entire ecosystem is balanced. But if the crickets start committing suicide, the food chain changes. So this is how simple movement of parasites can change the entire ecosystem. Now, in fact, in Africa, we have removed smallpox in our country. Similarly, there was a very bad disease which attacked herbivores, especially the ungulates. That's a rinderpest. And we have been able to eradicate rinderpest. Now, what has happened is that due to eradication of rinderpest in Serengeti National Park, plants have been grazed much more. So grass is being grazed much more efficiently. Therefore, forest fires have reduced. More trees have started growing. You see how the entire ecological cycle is getting affected because of removal of one of the parasites. Now, this is another kind of a parasitism which we all know, cuckoo and crow. That's nest parasitism. We call it also the brood parasitism. But I am going to talk of a different bird. Now, these examples are not mainly from India because we have not done such in-depth studies on these relationships. So wherever I got information, wherever I have worked some part, those examples I am giving you. Now, here is an example of the cuckoo finch of Africa. Now, this cuckoo finch is a bird which doesn't make its nest. It lays eggs in other birds' nests like the warblers or the prenias all over the Africa. So these are their host birds, and the cuckoo finch lays the eggs in their nest. And in fact, you can see the nest is so small and the chick is so big. But still the prenias and the warblers start feeding them because basically it is a fixed action pattern. It's an innate behavior that once it is started, the bird will continue to feed it, not looking that it is not his chick or her chick. So the cuckoo is taking advantage of this parenting behavior of these birds. Now, please remember, sometimes it is not always that the cuckoo is successful. Many times the egg laid by the cuckoo is understood as a foreign egg and the parent birds throw it out or destroys it. That happens. But the cuckoo, what it does is, it goes on laying there once. ठीक है, फेंक दिया ना, और एक ले. So by the time second, third egg is laid, the bird gets used to it. And then the cycle is started. Okay. And another thing what the cuckoo does it doesn't lay egg in one. Even our quail doesn't do that. It will lay egg in several crow's nests. Similarly, here also it will lay in several nests. And they all know कि कहाँ मैंने egg डाला है. So they always remain there कि कहाँ उधर उसको उठा के फेंका है तो again go and lay more egg. So they are always in the parent cuckoos are always in the vicinity. And they watch how the eggs are getting developed, how the chicks are being fed. In fact, there have been cases where the nests have been protected also. So unka chick nahi jana chahiye. So such a beautiful adaptations these cuckoos have for this brood parasitism. Now, another thing which will come into your mind is that the chick is listening to the foster parent sounds and songs, but it is supposed to sing like the cuckoo. So how does it do it? So for them, the imprinting of song is much later. So the hatching happens, the chick is fed, it comes out as a hatchling, still it is being fed by the foster parents. And then when it is really ready to fly and live on its own, the parent cuckoo starts coming and singing around it very loudly. And that is the time that whatever imprint that it has in his brain of the song, that gets, Arey haan, ye mera parents ka sound hai. Okay, now I will start remembering this. And it will start singing. So there is no confusion that foster parent ne khilaya, lekin main gaana mere parent ka hi gaunga. So this is how the relationship is so very intricate. in this kind of interaction. Now, I told you that the host many times will identify the the nest parasite egg. The hosts have learned to lay different types of eggs. Can you build it? This so many of these types of eggs. Whenever it goes the wobbler nest, it will lay egg like the wobbler. When it goes to prenia, it will lay like the prenier. And there have been females which have been found to lay eggs in 6 to 7 different species. This is a varied adaptation, which cannot be explained at any level unless we know the genetics of it. There is genetics in it. Now, what they have found is that the gene for this production of different types of egg is on the Y chromosome of the bird. That we see, you know that in birds it is ZW method. The reversal of the human's method of XX. XX is female in us and XY is male. In birds, the XY is female and XX is male. So the female carries the Y chromosome. And this making of different egg gene is present on the Y chromosome. So always the inheritance is from the mother. And this is another insurance because father mein do eggs hai. So in case there is some change in the mutation, the egg coloration technique or the ability will go off. So avoid that. The maternal inheritance is more existent in these birds. Okay. Here is a very completely different parasitism. The ladybird beetle. This is the red spot, the black spotted red ladybird beetle. It changes its behavior and it becomes a protector of eggs and cocoon after it is infected by this wasp. See this relationship. The wasp stings the ladybird beetle. Okay, so we start from the top. So the wasp stings the ladybird beetle and it releases those eggs into that abdomen. And then the egg starts growing, the larvae comes out, and larvae starts feeding on the internal organs of the beetle. Little. So the beetle is still live, it is moving around, but it is carrying the larvae, which is feeding on itself. And then it comes out from the back of the ladybird beetle and becomes a pupae. And then the pupae is held between the legs. The pupation actually happens between the legs of the ladybird. This fellow cannot move, but is still alive. And then it does something very, very surprising. It sits with all the legs covering the pupae and sits on top of the pupae. Because the ladybirds are very brightly colored, they have some alkaloids in them, so birds don't feed on them. So they are anyway not eaten. And this pupae can be eaten, but this is within the legs of the ladybird. So the pupae is also protected. To show that it is alive, intermittently this fellow will shiver very strongly to show that yes, I am alive, so that the birds will not come and touch the pupae. And this continues till the adult is coming out from the pupae. And then finally, the ladybird becomes normal and it starts life cycle again. And we have found that in some cases the wasp again infests the same ladybird. So sometimes the ladybird gets infested at least 3 times, but still it is surviving. Now one scientist started working on it a little more in detail. And he found out that the ladybird's head is full of a virus. So from where did this virus come? So he checked the wasp. And then he found out that the wasp was the one who was carrying the virus. So this virus, so when the wasp is laying the egg, it also releases the virus into the ladybird. And the virus is the one which changes the behavior of the ladybird beetle. Now the most important thing is that the immunity of the ladybird beetle is completely shut down by the virus. And then the larvae, when it emerges out, it wants the ladybird to be protector, So some signal is sent to the virus, and the virus reactivates the immunity of the ladybird, and the virus get killed, and the ladybird becomes active and becomes normal when the pupae is emerged into the adult. So now you can imagine there is now interaction of 3 different species: wasp, virus, and ladybird bee in this entire parasitic. This is something which we can't say there's co-evolution has happened. So all answers are from there. That's another enigma. These are the electron microscope. I'm not going to the detail because it's already 1:45. I'll just try to fast walk. Now this is some very interesting. The social parasites. Now social parasites are slightly different. Now these are normally seen in wasps and ants. Social parasites don't build nest. उनकी आराम की जिंदगी रहते हैं. But they want their young ones to be cared by somebody. So they colonize those host colonies. So what they do is they search for a colony. Either they enter the colony and remain there because they produce scent similar to the ants of the colony. So the ants don't realize कि ये कोई नया है. They start feeding them. Everything is given. Then these colonizing ants start laying the eggs. Those eggs are also taken by the workers. They care for it. So the young ones grow in the colony. There are some ants which attack the colony, picks up the larvae, hijacks them to their nest. And those larvae, when they come out, they believe that they are helping the adults of their own species. And these social parasites, they survive with larvae which has been brought from some other ant colonies. So this is another kind of parasitism which is seen. Now here you can see the social parasites have little bit of appearance similar to the queen of the colonized colony, and therefore that distinction is not understood. The workers consider both of them to be helped and maintained. Now if you see them very clearly in this photograph, you can see there are difference in colors. As human beings with our eyes we can array to dono alag dikhta hai. How are they not understanding? But ants survive on chemosignals. So these colonizing ants, they produce some signals which the other colony ants accept, and they consider that they are their own. So this is how they survive. Now here is a very classic example that can affect us. This is the Toxoplasma gondii, which is seen in the cat. So they sexually reproduce in the gut of the cats, all cats. So the parasite comes out from the feces from the cat, and they get into the food and water, and they contaminate it, then gets into the intermediate host, which can be any of them, cattle, birds, and sometimes even human. And then here is an example that if it gets into a rat, this Toxoplasma produces some chemical, and this fellow becomes very daring. It forgets its fear for the smell of cat's urine. And the rat will go and sit in front, come on, eat me. So this kind of behavior starts happening in the rat. Now, so this is how the Toxoplasma gets into the cat and the life cycle is completed. So by simply making it forget the risk of being attacked by a cat, it allows the rat to be eaten by the cat, and then the parasite, this protozoan parasite, enters the cat. Now, it has been found that even humans are also infected with this gondii. So the question was, why should we get infected? If you look back to our ancestry, we were also eaten by cats. So probably that remnant of that ability to infest the human beings is still there in gondii. And we have infested people all over the world. Everywhere there are people who get, especially those who have cats in their house, they generally get infected with these protozoans. Now, what is very interesting is some scientists worked on it. इसमें कुछ risk factor है ना, because the risk taking becomes more enhanced. So they checked all over the globe that are there any signs of taking risk to be increased in those human beings who are infested with this Gondai. And they found that they all are very daring. They start taking risks. And one of the risks, what they checked was entrepreneurship. So they found that in those places where there is high incidence of Gondai infection, startups are more, entrepreneurship is more, because they are taking risk, no? So now if you want to be a successful startup, get infected with Gondai. Now is it very easy to identify Gondai infection? Yes. Oh, ek strip milta hai, you use your saliva, put the strip, and you can check out whether there is an infection or not. So try it. But I have not yet subscribed to this theory that Gondai infection can make you really a good initiator for business, because you lose your fear for risk. But it definitely happens in mouse and all other animals. Probably also happened in our ancestors. But whether to link it with our business tendencies is something that needs little more scientific. But this paper is there. It's very popular paper. Many people have commented on it. But there is a trend. Statistically, there is a trend which shows that wherever there is a high Gondai infection, startups and entrepreneurships are much more in those areas of the world.
But this particular parasite even includes teratogen infections.
Yes, it does have. But the infection in human being is not so very high. So that is why we are saved that way. Now, the question is, do chemicals dictate our behavior? If it can happen in all those animals, we also have similar neurons. Most of our neurohormones are similar. Many of the neurochemicals that are passed from one neuron to the other are similar. So do chemicals dictate our behavior? So I will try to talk about that as the last part of my talk. I'll take the example of oxytocin in men and prolactin in men. We all know oxytocin is for production of milk and prolactin is for lactation. Now the question is, why should men have it? So did you ever think of that? We know that there is some function of this in men also, including women. Of course, we can very clearly relate it in the childbearing years, in the reproductive age, in women, why these hormones go up. But what happens to this hormone after the reproductive age in women? And what happens, and what are their role in men at the first place at all? So, yeah, but here it is something different. Here it is different. So, there is a paper where they have looked at romantic love and reproductive hormone in women. And they found, very interesting, women in love have higher levels of gonadotropin, FSH and LH, and lower free testosterone levels compared to single women. So there is a very clear-cut dictation of hormonal levels over our behavior. This is very clear. Now, what's very interesting is that this is a very interesting paper from Harvard where oxytocin has been classified as a love hormone. Now, what does it mean? It helps us to bond well with our loved ones. The more oxytocin, more we will bond with our loved ones. In fact, people have started making oxytocin sprays. Okay. Now, and people have found that if you are listening to your music that you love and you like to hear, your oxytocin levels are higher. So therefore, you feel it's a feel-good hormone. Now, there is low oxytocin after postpartum, and when they are in a low depression state, oxytocin levels are also low. One more thing that has been found, I told you this nasal spray is available of oxytocin, so you can change your mood. Now, oxytocin increases with exercise, obviously. exercise makes a lot of difference to a lot of chemicals, endorphins in our body. It also increases while with music and singing, and physical contacts, hugging, cuddling, all these increases oxytocin in our body. And that's why we love to be touched by someone whom we know very well, love very well. So these oxytocin levels are actually increasing, and that is why we feel good. And the newest thing is that if you get more of likes in your WhatsApp or your profiles, oxytocin is known to increase. So you see how the relation, even to the latest development in technology, they have related that oxytocin increases even in that. So the question is, agree? Thank you.
Had lecture of Sharma sir who said oxytocin is stimulated by chocolates also. That is why it will be Valentine's Day to check chocolate.
If you're not hungry, I can answer questions.
Any questions?
I was given 1 hour. It's okay.
Yes.
Any questions? Otherwise,
Students, it is a great opportunity to interact with sir. If you have any questions, please feel free to ask him. MSc, Bhagwat.
my only very honest request to you youngsters would be, these are areas which are very, glamorous to do research. And all these molecules that we were talking about that were making so much of difference are of therapeutic value. So companies have started looking at these peptides as commercial peptides for therapeutic. And huge amount of work is not being done in India at all. We really don't know any kind of these interactions. Recently, one forest officer had put a video on internet which became so viral about a bug being attacked by a fungus, and it was a zombie. He had taken the shots from all sides and showed that the bug is completely hollow, but still it is walking. So this is one instance that I got from India. But there are such kind of scenes are seen, but people have not really worked. For you all, working in this would be a great thing. You can do the physiology, you can do the biochemistry, and of course the molecular biology secrets of this big enigma. Thank you.
Yeah, more time. इसको कॉपी कर लेंगे
ऊपर से है। कुछ क्वेश्चन चाहिए नहीं? ठीक है। ठीक है। एक बंद कर
Yes. Thank you.
Thank you very much, sir, for that very interesting, very informative, and I truly believe it has become very infectious, sir. Sir's lecture, I think like the example sir has quoted, but I also believe, sir, there are positive impact as well. Where you have taken the control of the entire audience over here and have mesmerized with this talk. And I truly believe that our students have metamorphosized into learning more and becoming a true researchers in the coming future. Thank you so much. A big round of applause for sir. In the beginning, by slip of tongue, I used the word addiction, but now I feel that we are truly addicted. Addicted to the lecture that you have delivered, and we would want to have more lectures to hear from you. Thank you very much, sir. And I feel, yes, please. That's the behavioral change now. I think we can see in the audience. Thanks a lot. And I truly believe that, behold, I do not give lectures or a little charity. When I give myself. That's what Walt Whitman had said, and I truly believe that's what you have done today for our students and for all the teaching faculties over here. Thanks a lot, sir. May I now request Janvi Madam to kindly propose the vote of thanks, Madam.
Thank you, everyone. I am overwhelmed today to propose the vote of thanks on the behalf of Biological Science Department and ATBS, that is Association for Teachers. It's really an emotional moment for me, as many of the sir students will agree to this. Sir has just transformed lives. It is not just about his students, but any people who came in contact with him. He has transformed lives. That is what I would say about sir. It's just like a biochemical pathway, wherein there is an acquisition of energy, then transforming this energy and utilization of energy. Similarly, we have sourced knowledge from our sir. He has introduced us to the world of books, as Sachin Menon sir has already said. This same knowledge he has transformed us, and this knowledge, how to utilize this knowledge, that is what we have learned from our sir. So my heartfelt thanks to the director, Honorable Director Dr. Usha Mukundan, our Principal Dr. Himanshu Laura, our respected Vice Principals, Major Praveen Nayak sir, our Head of the Department, Mrs. Hanika Gupta, all the teachers and the faculty members of the Department of Zoology and Botany, all the dignitaries who are attending this session online, and my dear students, above all, you all. Thank you very much. Not but not the least, I would like to have our technical assistance, which is given by our staff members, Mr. Hemant Redekar and Mr. Sanjay Vishwakarma. As well as I would like to thank our non-teaching staff, Mr. Tahil and Mr. Sandesh. Thank you for your help. So once again, thank you everyone.
We can end the meeting at this time, right? The Zoom meeting. Participants who are present online, we are closing the Zoom